Okay. Well, good evening, everyone. Thanks for joining us this last webinar workshop. The series of 8 is coming to an end this evening. So it's been, hopefully it's been a useful couple of months if you've been participating in some of these as we've gone along. Tonight's topic is one that probably is one of the most important ones you might think, I suppose, trying to learn how to identify some of the common weeds and invasive species that we have to deal with, so many of them on our lands that we are trying to manage and protect. So I would just echo what Lance said. If you have any questions, be sure to put them into chat. We'll take a break if it looks like that'll work about halfway through. Can answer some questions then if there are any. And then again at the end. Again, these, as Lance said, these are going to be available, have been available as videos. You can always go back and review them, or if you didn't get a chance to join us tonight, you can do that. The handouts are only available to those who register, though. So that's one advantage of doing that. All right, so I start now here with 4 species here that are invasive species. These were 4 species that were identified by a Smithsonian article back in 2013, so it's a little bit old, but they were pointing out the worst or most threatening invasive species in the United States, and they were talking about the top 6 invasive species. These are the top 4 that they identified. This one up here is purple loosestrife up in the upper left. That was the first most important one. Then Japanese honeysuckle over here on the upper right was number 2. Japanese barberry over here on the lower left was number 3, and Norway maple was number 4. The other 2 species were English ivy and kudzu. Now, I don't know who wrote the article for sure, and of course it was based on the United States, so it may not be real reflective, of course, of what we deal with here. And in fact, we don't have any problems with kudzu, at least not yet, thank goodness. We don't have any problems with English ivy. We don't really have any problems with Norway maple or Japanese honeysuckle for that matter. So 4 of the top 6 are not problems for us. I guess that's good. But we have plenty of others to worry about. More than our share, probably. So just to set the stage here, I just threw some definitions of what an invasive species is. So we're kind of on the same page there. Most definitions do agree that it's usually a non-native species, but they put it in the context of non-native to the ecosystem that you're thinking about or that you're working with. And of course, most of them are indeed non-native to North America. But there's an important point there in that, especially in the second one, is that it doesn't have to be non-native to North America to be an invasive species. Species is given a status based on its origin, and if it's native to North America, then that's the case. If it's not native to North America, it's coming from somewhere else. But of course, within North America, species are native to can be native to just certain parts of North America. And a good example that they suggested here was that the lake trout is a species of fish, of course, native to the Great Lakes, but they've been introduced to Yellowstone Lake out in Yellowstone National Park, and they're considered to be an invasive species out there because it's a different ecosystem, of course, and they're not native to that. We have our own species, of course, is a good example of that in eastern red cedar. Well, it's even a little bit more different. Eastern red cedar is, of course, native to all of Iowa, and it's even native to prairies, but its population size typically on prairies, at least on the native landscape, was always pretty small due to the occurrence of fire. And now with over 100 years of fire suppression, many places, especially in the Los Hills and in the southern part of the state, and to some extent, even in some of the hill prairies in northeast Iowa, eastern red cedar certainly qualifies as an invasive species there, because it is doing exactly what this is saying, causing environmental harm. And that's the one that, again, we're mostly concerned with. But note that it doesn't have to be, just it doesn't have to be a species that's doing ecological damage per se. It could be a species that's harming human health too, and that would make it qualify. So just three definitions. They're all pretty much saying the same thing. They usually are characterized by, again, an ability to spread aggressively. In other words, to increase population size. Most of the time, we think that that's probably because the fact that they're not native to that ecosystem or to that place where they haven't evolved with the same set of species. Of course, they're new. They're a novel species in an ecosystem where all the native species have evolved interactions with each other that provide some population regulation. The populations that are native to the system have interactions that intend to limit the sizes of those populations. When a new species comes in that comes from some other part of the world, it doesn't have those connections with species, and so there's often a problem there in that there's nothing that's really there that's capable of controlling the population growth. So I looked up on, this was www.invasive.org, a website that does try to provide at least a count of the numbers of invasive species. The United States Geological Survey, USGS, has an invasive species program, and they say that there's more than 6,500 non-indigenous species that are now established in the United States. 6,500 non-native species. Now, again, not all of those are invasive, of course. Lots and lots of non-native species are present, of course, and they're not invasive. Thank goodness that they're not all. We'd really be having a big battle. But invasives.org, again, using the definitions that we've provided here, 1,800 plants in the United States or North America. I think this is mainly the United States, so just the 50 states. Most of those are forbs. As you can see, over almost half of them are forbs, not quite. Shrubs are the next most abundant, and then grasses and sedge-like plants. In the animal kingdom, only about, less than half, 750 or so. And not surprisingly, insects are take the lead by a long ways there. Almost 500 different species of insect that are invasive. And then the last group are pathogens, which is primarily fungi that cause disease in trees or other plants. So I did some checking with these three websites, ISU Extension and Midwest Invasive Species Network and Iowa DNR. They all have websites that talk about invasive species in Iowa. And by just putting together the species that they identify as invasive in Iowa, I came up with this list of 56 species. And of course, it's not complete. There's certainly more than that, but this might be, again, some of the top most important ones. The Midwest Invasive Species Information Network, they qualified the list with some classes or categories. So the species that are in blue font there are, I think, they are identified as primary noxious weeds. So again, that's a category that's given to a species by the state. And of course, a primary noxious weed would be one that is considered to be the biggest problem or causing the most problems and needs to be controlled. The red ones are secondary noxious weeds. Then the ones that are asterisk there, the Midwest Invasive Species website identified those as prohibited. So I'm assuming that means that they're not available for sale anymore, at least. They probably were, but not anymore. So 56 species here. We're going to take a look at 30 species tonight. That's probably enough for us to try to handle in one evening. And many of them are on this list. There's some additional species that I'm going to be talking about that aren't on this list. So again, there's probably there's still probably several more that need to be on this list. So we'll start out with some legumes or Fabaceae species. And what I'm going to try to do is just try to give you enough information to help you identify, and especially separate these species from other similar looking species, and trying to do it to some extent when they're vegetative. Because again, in order to control these species, being able to identify them prior to flowering is certainly a big help. So the first two here are white sweet clover and yellow sweet clover. I'm going to put them together in a group there because they're so similar. And in fact, I've heard that when Flora of North America comes out with the Fabaceae, that they may end up being combined together as the same species. They're not that way in Flora, Missouri, but we'll see, I guess. And then another species that's pretty similar to the sweet clovers is alfalfa, Metacago sativa. Now it's probably not quite as invasive as Mellolotus, as the sweet clovers, but it certainly does occur somewhat, especially in prairie restorations where alfalfa may have been a species that was present at one time. So in all of these legumes, all of these Fabaceae are going to have trifoliate leaves. So trifoliate means that they're compound with 3 leaflets, like you see in all of these examples here. The term trifoliate is used as a general term to, again, identify any compound leaf with 3 leaflets. We can also say that this one, and I think this one down here too, are pinnately trifoliate, because the petiolule, which is the stem that we see right here for the terminal leaflet, that's longer than the petiolules or the stems for the 2 lower leaflets. So that would make this a pinnately trifoliate leaf. And most of the Fabaceae are like that. Now, the way to separate, if you don't have flowers, obviously you have flowers, you can easily identify white and yellow sweet clover by the flowers. If you don't have the flowers, you're not going to be able to tell which of the 2 sweet clovers you've got. Vegetatively, white and yellow are so close to each other, it's just not possible. Either of them, of course, have a different colored flower than alfalfa, which is bluish. So again, if you have flowers, there's really not too much of a problem. But without flowers, you're just looking at leaflets. And probably the best thing here is that in sweet clover, the leaflets have teeth pretty much all the way around. Starts right here on this one, tiny little teeth all the way around. So we would say the leaflets are serrated or have teeth on their margins for the entire margin. In the alfalfa, Metacago, they're just on the outer 1/2 to 1/3. So the teeth are only present about right out in here. Again, they're pretty hard to see because they're really small teeth. If you're holding this in your hand, of course, you can probably see them without too much trouble. A hand lens might be some help. So that's probably the best way. The leaf shape, well, excuse me, the leaflet shape is pretty much the same. So that doesn't really help much at all. It's the serrations that are going to help you there. Another thing that you can do, and I do this a lot, is start to learn how plants smell. Sweet clover has a particular smell that a lot of times if I see something vegetatively, I think, oh, I think that's sweet clover. I'll just grab a leaf or 2 and crush it and smell it. And yep, that's what it is, just to be sure. And alfalfa has its own smell too. They don't smell the same. They have a different smell. Mellolotus has coumarin in it, which gives it sort of a sweet scent. You might say it was kind of resembling the smell of vanilla. I'd say it's a little bit different than that, but there might be a touch of it there. And alfalfa doesn't have any coumarin in it, so it has a different smell. It's something I really can't describe to you very well. It's something you just have to teach yourself by taking the time to, well, when you, especially when it's in flower, you know exactly what it is because the flowers are there. And go ahead and you know, grab a few leaves or leaflets and crush them up a little bit and see what they smell like. I put this is the underside of alfalfa leaf, which shows quite a bit of pubescence, but basically both of these species are variously glabrous to pubescent. The amount of pubescence varies. Somewhat more pubescence on the underside in both species, less pubescence on the top side, but the pubescence is not going to help very much. All right, next one. Two more Fabaceae. Now, these two are pretty important from the standpoint of prairie management. Black medic, which is Medicago lupulina, and bird's foot trefoil, which is Lotus caniculatus. Both of these are pretty different, of course, when you're looking at the leaves here. Black medic is, again, an example of a trifoliate, but it's pinnately trifoliate because again, the petiolule here is longer than the petiolule here. In fact, these leaflets are pretty much sessile. And what bird's foot trefoil has is a leaflet that has a leaf that has five leaflets. It's got the three leaflets that are towards the end of the leaf, these three up here, and it's got two leaflets right down at the base of the leaf, so that the leaf is basically sessile. There really isn't any petiole here because these two leaflets right here would mark the bottom of the leaf, and that's where the leaf is attached to the stem. The leaflets, of course, are quite differently shaped. Bird's foot trefoil, all you need to do is, again, take a close look, and if you can see that there's five leaflets, three of them at one end of the leaf, these are all pretty well clustered together, are basically palmately trifoliate. These three are palmately trifoliate because the petiolules, the little leaves, the leaflet stalks are all the same size. And then it has two other leaflets that are at the base, bird's foot trefoil. Again, if you have the flowers, then it's a lot easier. Of course, it's always going to be easier with the flowers. Some interpretations, you might see some interpretations of bird's foot trefoil that describe this as being a palmately trifoliate leaf with three leaflets and then having two leaf-like stipules. That'd be one way of interpreting what's happening here, but I've checked several sources and they all agree that's not the correct interpretation. These are not leaf-like stipules. I kind of makes you wonder though, because there are no stipules in bird's foot trefoil. There's basically a couple of minute little gland-like dots that are present at the base of at the end of the leaf down in here, which some interpret as highly modified stipules, but they don't really seem like, you know, that wouldn't be a good example of what a stipule is. So it kind of seems to me that they don't have stipules. So, but almost all Fabaceae do have stipules. Almost all of them do. So I kind of wonder if that interpretation is actually right, and if maybe these really are stipules. But another thing to point out here is the venation pattern. Venation pattern in Medicago is pretty conspicuous. You can see it, pretty well here. You can always usually see it a little bit better on the underside of the leaf. And not all of these leaflets show this characteristic, but this is a characteristic that is used. There's a tiny little, what's called a mucro, which is a tiny little point. There's a tiny little point at the tip of each of these leaflets. It's basically a little bit of an extension of the midvein that comes out past the margin of the leaf just a little bit. So you can kind of see it right there as well. It's really, really small. And I really don't see any on this one here, so it's not always present. There's one on this leaflet right up here at the top. There's one right there. So the thing that you're going to confuse this most likely with would be white clover. And white clover does not have a pinnately trifoliate leaf. White clover has a palmately trifoliate leaf. In other words, the 3 leaflets in white clover all have the same length of petiolule. Again, petiolule is a term I'm using to describe the little stalk of the leaflet that attaches the leaflet to the rachis, which is the central part of the compound leaf, this thing here. Next two are, oops, I'm going the wrong way, sorry. Cow vetch and crown vetch. Well, crown vetch is really a bad invasive species, one of our worst ones. And cow vetch is becoming more invasive, it seems like. So again, two that are pretty important. They're somewhat similar in having pretty large compound, pinnately compound leaves with quite a few leaflets. And I'm also going to compare crown vetch with a native species that it can be somewhat similar looking to. Again, if you don't have flowers, if you have flowers, then there's no problem telling Canada milk vetch, which has these yellow flowers, you can kind of see a little bit of right here. No problem separating it from crown vetch, which these really faint lavender to slightly purplish flowers are. But if you don't have the flowers, you just have vegetative growth, then you're dealing just with the leaves, then it's not so easy, especially if the Canada milk vetch is a fairly short plant. Because again, if it's a short plant, it's going to look more like crown vetch. Well, so the way to separate crown vetch from Canada milk vetch is that these compound leaves here, this is crown vetch, have leaflets that go all the way down to the base of the leaf. So that again, in essence, there is virtually no petiole. The compound leaf is basically sessile. It can be a tiny little petiole there, basically, but nothing like we see over here. This is a compound leaf from Canada milk vetch. These are the last 2 leaflets right here, and so all of this right in here is a petiole, and it's basically 10 to 30 millimeters or so long, so 1 to 3 centimeters in length. The other thing are the stipules. And so here are the stipules of Canada milk vetch. Canada milk vetch is inside this pink border here. Pretty large and conspicuous stipules. The stipules are anywhere from 4 to 18 millimeters long and pretty broadly lancelet shaped. This one's a little bit smaller on this side over here. Remember, stipules are 2 small paired sort of leaf-like, bract-like structures that are attached to the leaf, but right at the bottom of the petiole, or at the base of the leaf. The stipules on crown vetch are shown right here. You can see one of them right here, and we see another one right here. So the stipules on crown vetch are much smaller. They're only about 1 to 4 millimeters long. So clearly do not overlap with the stipules we see here in terms of their length. And these are much more narrowly lance shaped. So that'd be a good way to separate crown vetch from Canada milk vetch. For any of the vetches that are in the genus Lathyrus, and this is well, excuse me. Well, actually, this is Vixia. So this is actually wrong here. I thought I was going to get a Lathyrus as an example here, but I couldn't find a good one, I guess. So this is Vixia cracca, cow vetch. So Vixias and Lathyrus, both genera have tendrils. This compound leaf here, the end of it, the end of the rachis, this is the rachis, the central part of the compound leaf, the end of the rachis continues on and forms a tendril. A tendril is a structure that wraps around things and hangs on to things as to help the plant be a vine. And here's an example of tendrils over here as well. Cow vetch in particular is also pretty pubescent. But if you see, again, tendrils on a compound leaf, then you've got either Lathyrus or Vixia. Both of them are types of either a vetch or vetchling. All right, the next two are the clovers, white clover, Trifolium repens, and red clover, Trifolium pratense. So here again, we see, again, when they're in flower, no problem at all, different colored flowers. And really not too much of a problem if they're in just vegetative form either. Trifolium repens, white clover, one thing you should know about it, of course, is that it has horizontal stems. Basically, what you see here are all just leaves that are coming up from a horizontal stem. Basically, a stolon is a horizontal stem that runs on the top of the ground. And that's what happens with white clovers. They have these stolons that are spreading out, and then pretty long petiolate leaves that we see here. You don't see the petioles. I'm just showing the leaflets here, but the petioles of these compound leaves is pretty long and attaches to the stolon. So again, here we have a palmately trifoliate leaf. And again, with white clover, the leaflets are pretty orbicular, pretty much circular. They can be somewhat suborbicular. They can be a little bit more like red clover. In other words, just a little bit more, you might say, orbiculate to elliptic or so in shape, or to obovate. The veins, again, are pretty conspicuous in white clover as well. And in red clover, there's often this chevron, this watermark here that we see, this inverted V basically. And that same kind of chevron can be present in white clover too. It's not present in the leaflets as often as it is in red clover. I threw just a picture of the flowers in just to remind you that, again, all of these Fabaceae that we've been talking about have the, this is called a paplinoid type of flower, the basic P type of flower, which is composed of a large banner petal, this large banner petal, which is kind of behind the rest of the flower and kind of above the rest of the flower. Then there's a couple of petals down here that are called the wings. And then there's a couple of petals that are fused together to form this structure called a keel. And the stamens and the pistil are inside that keel. But these paplinoid flowers, again, are common in all of these plants that we're seeing here. So I think, again, there's the stipules on red clover. They're pretty distinctive. They're partially fused together on one side, but pretty conspicuous. And another thing that's important, persistent. Persistent means they stay there. They don't fall off. All right, I think we've got two more Fabaceae, and then we'll move on to a different group. Here we've got one of the really bad members of the Fabaceae when it comes to invasiveness, Sericea lespedeza cuneata. And so it's going to look like, it doesn't really look too much like round-headed bush clover, which is shown right here, again in the pink border here. That's round-headed bush clover. The leaflets resemble a little bit. Sericea lespedeza looks more like Lespedeza virginica. But it's really distinctive in a couple of ways. And again, it's a really important one because it's so invasive. One way is that the stems have these very clear and somewhat distinctive lines of pubescence. Now, other Lespedeza can have pubescence on the stems, but it's usually more scattered. Here again, we can see pretty distinctive lines of hairs running vertically up and down the stem. And the other thing that's distinctive, in fact, no other native Lespedeza has this, the venation pattern that we're seeing here on the adaxial side, which is the top side of a leaf of Sericea. We can see the veins here, and the veins are the term that's used to describe what we're seeing here in terms of how this venation appears to us. It's called herringbone venation. Herringbone venation is when the veins come off of a main vein like this, the side veins come off, and they kind of come off in a regular kind of parallel fashion, and they run straight to the margin of the leaf without branching or doing anything else. Here we see in round-headed bush clover, this is not herringbone venation. All of the native Lespedezas have venation that looks more like this. We can see that the lateral veins come off, but then they interconnect with other veins. They branch and they interconnect in a kind of a complex way. In Sericea, again, that doesn't happen. The veins go straight to the margin without branching. And again, sort of parallel to each other and at a regular spacing. It's kind of like the bones in a fish. That's where the name herringbone venation comes from. Then the other one down here is black locust, another really bad member of the Fabaceae when it comes to invasiveness. It can be really bad in some of our drier sand prairies. Well, the thing that really, again, helps identify black locust or Robinia pseudoacacia are the two spines that most of the time are present, maybe not always, but most of the time. These two spines that we see here, they're paired. This is on some new growth. These two spines are on some older growth. So you see they don't look quite the same there, but they're still there. These two spines are modified stipules. Since they're modified stipules, they're formed from a part of a leaf. Anything that becomes sharp and pointy and originates from a leaf is called a spine. The flowers on black locust, they produce these really big papillinoid types of flowers, really large, a large raceme of flowers. The leaves themselves are usually just one spinnately compound. The only thing that you might kind of confuse this with, of course, would be honey locust, probably. Honey locust doesn't have spines. Honey locust has thorns, so that's an important difference. And honey locust has much smaller leaflets than these, if you were to compare them side by side. And quite often, honey locust will have at least some leaves, especially on some of the older branches. The larger leaves will be twice pinnately compound rather than once. This is called one pinnately compound because the leaflets are attached immediately to the rachis. The rachis is an extension of the petiole, this part right here. And since the leaflets are attached to it, this is called the primary rachis because it's the only one. It's the first one. And if the leaflets are attached to the primary rachis, then your leaf is once pinnately compound. All right, we're going to move into a different family now and look at something in the Apiaceae family, the parsley family. A couple of bad players here. The first one is up here is poison hemlock, Conia maculatum, and the one below here is wild carrot or Docus carota. So they're both members of the parsley family. So they both have a very important characteristic that almost all members of this family have, and that's this inflorescence that's called a compound umbel. The flowers on wild parsley, anything in the parsley family, and on hemlock and wild carrot, and all of the other ones, things like golden Alexanders and water hemlock, a couple of native species, the flowers are really, really small. Tiny, tiny little flowers. In this diagram, a flower is represented by each of these little circles. That's a single flower. The flowers are really small, and what happens is in this inflorescence, they get aggregated together in a way that the inflorescence looks like a larger single flower. Same thing kind of happens in the Asteraceae, as you know, lots and lots of small little flowers are aggregated together into a group to make it look like a larger flower. Well, in the Apiaceae, though, is a particularly important way that these are aggregated together. So on these little tiny flowers, most of the time, flowers have little flower stalks, a little stalk that attaches a flower to whatever it's attached to. That little flower stalk is called a pedicel. And so what we see up here are a group of flowers with their pedicels all attached to a common point. There's a couple of, well, there's 3 bracts. There's 3 bracts that subtend this umbellet. So these little groups of flowers are umbels, because that's what we call a flower structure that has the flowers, the pedicels all attached to a common point. The term for that is an umbel. Since these are really small umbels and they are a part of a compound umbel, the term umbellet, meaning a small one, is used to refer to each of these. Then each umbellet has a structure, a stem, if you will, these are called rays. And all of these rays connect all of these umbellets to a common point. So it's another umbel, a bigger one, and that's what a compound umbel is. Lots of small umbels aggregated together within a larger one. So again, almost all of the parsley or wild carrot family have compound umbels. This one right here actually looks very similar to the one for wild carrot down here. The compound umbel structure has some bracts associated with it right here too. These bracts form an involucre that again subtends the largest compound one. Again, these bracts in here make a smaller, similar kind of thing. It's called an involucre, since it's smaller, that subtend the umbellet. So both of these, again, have compound umbels, and both of these have tiny, tiny flowers that have corollas that are white. So they have a similar look to them. But as you can see, the stems are vastly different in terms of the poison hemlock having a glaber stem with some purple splotches and mottling. That's a good characteristic for it. Wild carrot always has some pubescence of some kind, sparse hairs here, but fairly long hairs. The leaves are usually bigger in poison hemlock and can be anywhere from 3 to 4 times compound. This one right here, I would say, looks like it is, so this is the primary rachis, and then a secondary rachis. And then it looks like these are separate. We have a tertiary rachis right in here. So this one is 3 times compound. They're usually 3 to 4 times compound, the ones that are really large. Some of the smaller leaves up higher on the plant are just 2 to 3 times. Wild carrot leaves are usually just 2 to 3 times. This one here is twice compound. The primary rachis is right here again, the secondary rachis is right here, and the leaflets are attached to it. This is a leaflet here. This is a leaflet. Up here, this is a leaflet. And in both these cases, the leaflets are also lobed. So there's also lobing that's present within the leaflet. This is just a close-up of the leaflets here on poison hemlock. And then I think the last thing is just that in wild carrot, what happens when this thing becomes more mature, the flowers are maturing and the fruits are forming. Here's a fruit of wild carrot, is that you see this happening. The rays, which again are these things here, these rays curve upward and kind of press in on the entire flower head and kind of form this ball, I guess you might say, that we're seeing here. Right in here. And the fruits on the Apiaceae or the parsley family, they're called schizocarps, and I'm not going to try and explain what those are right now, but they basically they do come apart. They come apart in a way that's related to the carpels that form them. But the point here is that the fruits have several ridges that are kind of winged, and these winged ridges have a series of bristles on them. These bristles here all have short little hooks that are present at the very end of the bristle. Hence, it's a fruit type that's going to be easily attaching to fur or to clothing. Any questions there? I don't think we have time maybe to take a break, so I'm just going to go ahead and push on through, I guess, get through the rest of these, and we can just take questions at the end. This one won't take too long. We talked about thistles last week, and so but some of you may not have been present, so I wanted to throw two of the most invasive thistles in here. We've got musk thistle at the top and bolt thistle on the bottom. And again, when they're in flower, not too much of a problem, because musk thistle has just single flowers that terminate branches. And quite often these flowers, as they really begin to mature and are ready to start dispersing their fruits, the flowers will tip over and start pointing downward. In other words, they'll nod, start to point downward. Bolt thistle has clusters of flowers at the top of branches because there's some short branches that come in there and make more of a group type of flower cluster. Both of them have, and this is important just to, in terms of vegetatively, both of them have these stems that have a wing structure that goes up and down the stem between the nodes. So it's these wings, you kind of see them both here, are going between the node of one leaf down to the node of another leaf. And these wings have little prickles, pretty large prickles actually, that are present along those wing stems. But vegetatively, if you don't have any of this, you don't have the flowers, you just have these basal rosettes, or maybe just some stem leaves, then what you need to pay attention to is that musk thistle always seems to have a much more shiny leaf surface. And these basal rosettes, and then this is a leaf that's on the stem. So if you just had some leaves on the stem, but you didn't have any flowers, you kind of see how shiny looking these leaves are compared to bolt thistle down here. Bolt thistle has leaves that are more dull looking. It's more of a dull greenish, I would say a little bit of a slight hint of sort of grayish green, more of a dull green look to them. And just looking at these two basal rosettes, you can see that the margins here on musk thistle are are lobed in a different way, I guess is the best way of saying it, are lobed in a much different way than the leaves down here. These are not lobed as deeply. These sinuses, which are the places that are between lobes where there isn't any leaf tissue, the sinuses don't cut into the leaf quite as far as they do down here. So that should help if you're trying to figure out which one you got and you don't have any flowers. A couple more another thing is just to remind you is that if you find a thistle and you look on the bottom surface of the leaf, which you should always do, and you see the bottom surface of the leaf is very white with dense, short hairs causing that whiteness, then you've got one of 4 native thistle species. It's either tall thistle, Cirsium altissimum, or field thistle, Cirsium discolor, or floodman's thistle, Cirsium floodmani, or wavy leaf thistle, Cirsium undulatum. Those 4 native thistles are all going to have a very white, clearly white surface on the bottom side of the leaf, which will contrast with a much greener surface on top. Okay, 2 more Asteraceae then are the Canna Thistle, which of course we all know is a really bad invasive species. And then I put spotted knapweed in here because it is in the same family. I don't see it as much as a problem, but last week, Leigh O'Neill reported that they do have a problem with spotted knapweed at Chautauqua. So it should be something we're on the watch out for. So with Canna Thistle, you're looking at a thistle that's going to be really, in most cases, very clonal, and it's very rhizomatous, and so there's going to be a lot of stems. Generally, if you have any Canna Thistles at all, there are going to be a lot of them growing closely together. Whereas again, these 2 species are biennials, and they're not going to be in any kind of dense patch, and they're going to be a lot taller as well. So Canna Thistle is always pretty densely populated in a patch. Here's what the leaves look like. They can have some white pubescence on the undersurface of the leaf, usually not nearly as much as those native thistles do. They have really small flower heads. That's probably one of the most characteristic things. You see quite a few flower heads over here, flower heads terminating many of the branches at the top of the plant. Here's a single flower head. We're zooming in on it. And we can see that the involucral bracts, that's all of these little structures here, all of the plants in the sunflower family or the Asteraceae have involucres, which are the place where there's usually several series of bracts that sit below the disc flowers. And of course, in both of all 4 of these species, the 2 thistles previously, and then this thistle, and then also knapweed, we have a type of Asteraceae here that has just disc flowers. There are no ray flowers, the big ray flowers that you would see on a sunflower, for example. They're not present here. And so these involucral bracts, they sit down below, they attach on the receptacle that again is the place where the disc flowers are attached up here. So on Canna Thistle, the involucral bracts have tiny little short spine tips, very, very short. On many of the, well, especially both thistle here, you can see these involucral bracts, they have gigantic spines, really sharp spines. And actually in musk thistle, the involucral bracts don't really have much of a spine at all. That's another way to tell them apart if you just had the flower, for example. These involucral bracts right here don't have much of a spine at all, just a short little spine tip, but they're very large bracts. These bracts here are pretty small, and then they just have a really small spine tip. Now, knapweeds are very different from the thistles. Some centaureos are called thistles. That's the common name. Some of them are called knapweeds, some are called thistles. The most important way that they're different, there's 2 ways that they're different, or maybe really 3, is that the involucral bracts in knapweeds don't have really any kind of a spine like most thistles do. They have just a little fringe of little ciliate hairs, so to speak. You kind of see those that fringe that surrounds each of these involucral bracts. In this particular species, spotted knapweed, the involucral bracts have this brown tip, and the fringe, the cilia, if you want to call them that, row of hairs or bristles. Bristles is probably a better term. Pretty much occur throughout most of the top part of that bract. So they don't have the spines again. And the other thing is, if you look at the fruits, I don't have a fruit of a thistle up here, but in a thistle, such as Circeum up here, the fruit, which is shown here, is completely symmetrical at the base. Both sides of the fruit are going to be just exactly the same. But in knapweeds, we see it's asymmetrical. There's this scar basically right here. This is a scar and this is a scar. That's a scar that's coming from where the ovary and what becomes the fruit, again, the ovary becomes the fruit. These are the fruits now, where the ovary and where the fruit was attached. Now these are detached, of course, and they detached. They were attached sort of obliquely to the receptacle. And that's why this scar is present here and visible kind of on the side. Then the other thing I would mention is knapweeds also have they have disc corollas, excuse me, they have disc flowers, but the disc flowers that are on the outer edge of the head have really large corolla lobes. These are the corolla lobes of those disc flowers, and they're really long, much longer than disc flower corolla lobes usually are. Okay, so that's knapweed, spotted knapweed. Now let's go on to a couple of species in the Polygonaceae. The worst one here certainly is the bottom one here, Japanese knotweed. It's terribly bad. This one up here, curly sourdock. It can be pretty invasive in some places, especially in damp soil. Well, but they're both in the Polygonaceae, of course, and one of the things that unites them that way, in a sense, is that they both have a structure called an ochrea, O-C-R-E-A, ochrea. In the Rumex up here in sourdock, the ochrea is seen right here. And what the ochrea is basically stipules that have joined and fused together to form a short little sheath that surrounds the stem right above where the leaf is attached. So this is an ochrea here, and this is an ochrea down here. Leaf is attached right here at this node. We can clearly see this is a node. Leaf's attached at the node, and the ochrea is then this little short sheath-like structure, a membrane type of structure that starts at the node and goes up the stem a ways. So that unites them. Another thing that kind of unites them is that in the flowers just have tepals. They've got sepals and petals, but the sepals and petals are very similar to each other. There's no difference really in them and how they look. And so when sepals and petals are basically identical, we use the term tepals, T-E-P-A-L-S. And in both cases, these have tepals. In Rumex up here, there are 6 tepals. There are 3 inner ones and 3 outer ones. The 3 inner ones get really large in fruit and enclose the fruit, which is an achene. So inside of this structure here is where you would find this. It's inside of this. And so there's 3 tepals here, so this whole structure is sort of 3-sided. On all 3 of these tepals that form this structure, there will be this callicite or tubercle. It's a kind of a fibrous, I guess you might describe it sort of a fibrous, some fibrous tissue. It basically forms this thing that we can call a callicite or a tubercle. There's one of these on each of the 3 sides, on each of the tepals. They form from the mid vein of that tepal. But in curly sourdock, now again, these things are common in all Rumex species, but in Rumex crispus, there's there's usually 3 of them, but one of them is always bigger than the other 2. So that's, and I can't show you that here, but there's going to be 3 of them here again, one on each of these 3 tepals. One of them is bigger than the other 2. Here we can see a whole bunch of them here. These tepals aren't quite mature yet. They'll turn brown like this when the fruit inside is more ready for dispersal. It's thought that these tubercles help in dispersing the fruits by helping the fruits, helping this whole structure really to float. All right, so that, and the other thing, of course, that's really important with Rumex crispus, its scientific name crispus, is in reference to the margins of the leaves. And you can see this basal rosette here. You can see this leaf is on a stem. You can see some leaves over here on the stem. The margins of these leaves, which are pretty large leaves, they're pretty long, fairly broad. The margin is always undulate or wavy, or sort of curled. And crispus is the term that's used then to describe sort of the condition of the margin of those leaves. That's a really good characteristic for identifying this species if you don't have any flowers or fruits. Those wavy leafed, or those wavy margins, crisp margin along the leaves. All right, Japanese knotweed again has the same ochrea. It has a little bit different structure to the flowers. There's only 5 tepals here rather than 6, and they do enlarge. There's 3 on the outside, 2 on the inside. All of them kind of enlarge somewhat, and again surround the fruit that's inside of here, which again is an achene, looks something like that. What's really distinctive here are the way the flowers are arranged in these big clusters with lots of branches to them, several branches in each of these clusters, all with a whole bunch of these flowers. And then of course their leaves. These are very characteristic leaves, very large leaves. They've got a very truncate base, which means that the base is squared off and is basically level across the base here. It's like a line going across here. And then again, just their size. Japanese knotweed is a fairly large plant, can get way over 2 meters tall, maybe even at least 3 meters tall, and it can form really dense patches, which again is one of the problems and why it's a very bad invasive species. Well, we better keep going so we get through here. Decent time. We've got a couple of honeysuckles here. I don't know why honeysuckles weren't on that list from the Smithsonian, but they're certainly among the most invasive species that we have. There's 2 really common ones, Amur honeysuckle, Lonicera mackii, and then Tatarian honeysuckle, Lonicera taterica. And there's a real straightforward difference in their leaves here. They're going to have simple opposite leaves, as we see here in this example, you see down here. But Amur honeysuckle always has, the leaves have these acuminate, what's called acuminate tips. Here we can see a couple of them a little better, and these aren't quite as acuminate as they sometimes are, but acuminate means that the tip of the leaf is really drawn out, and it is sort of a concave margin right through here. It's not quite as concave as it sometimes is, but the leaf tip is kind of like stretched out and drawn out to a very clear point. The point is clearly an acute point here. Down here, we don't have that acuminate shape to the leaf tip. I mean, it certainly tapers down to a point. The point is more broadly acute. In some cases, it may be closer to obtuse, but clear difference in the shape of the leaves here. There's also a clear difference in how the flowers are arranged. In the honeysuckles, we see an example right here for Tatarian, and we see an example here for Amur. There's two stalks basically in the axils of the paired leaves. So here's a leaf and here's a leaf. They're opposite each other, again, as they always are. In the axil of this leaf, there's a peduncle here that has two flowers attached to it. And then there's another peduncle over here that has two flowers attached to it. But the important thing here is that in Titterian, these peduncles are really long, very long. The peduncles in Amur are really short. And again, it's kind of a relative thing, but you can easily see it. And you'll see it with the fruits too, because of course the ovaries, what you see right here, that's what's going to become the fruit. So when you see the fruits are always much closer to the leaves, basically, in this species. Here we do see some fruits here that are further away from the axil of the leaf here, because again of that long peduncle. A couple other things: honeysuckles, the exotic honeysuckles, the non-native honeysuckles, one way they're always different from the native honeysuckles is what you see right here. If you were to take a cross section of a twig or maybe a 2 to 3 year old branch, you see that the center of the twig is hollow. So the very center doesn't have any tissue at all. And that hollow area is surrounded by some brown pith. So pith is the tissue that's usually in the center and fills up the center. Here, we could also say that the pith has a hole in it, I guess you might say. There is a little bit of pith tissue here that surrounds the edges of that hollow core. Now I put this leaf on here because I thought one plant that you can confuse honeysuckle with possibly is dogwoods. So again, in the pink border are two examples of dogwoods. Now in honeysuckle, the venation is different than here. In dogwoods, the venation looks like, well, I remember this is just a picture of the hairiness that's present on a Amur honeysuckle. And down here, this is showing the leaf, the bottom side of the leaf of Titterian honeysuckle. Titterian is almost always glabrous. There's no hairs hardly at all on it, whereas Amur honeysuckle has at least some hairs. Sometimes they're mainly just along the edge of the leaf, like you can see here, but there's always at least some hairs. Okay, no, so back to this. I'm gonna trace a vein here. The vein in a dogwood goes like that. This is a lateral vein. It comes off the mid vein here, and it goes towards the edge, but then it follows the edge of the leaf for quite a ways. That's called arcuate or arcing venation. That does not happen in the honeysuckles. It's hard to see those veins, but they basically just go out towards the edge and then just kind of stop. Another thing you can always check is the honey that separates the honeysuckles from the dogwoods. If you pull the leaf of a dogwood apart, as had been done here, there will be silky threads that you'll be able to see in the gap where the leaf has been torn. You can see all these silky threads here. Those silky threads are the vascular tissue that's joining the torn vein here and the torn vein here. So that's vascular tissue. Those silky threads are vascular tissue that is sort of elastic and doesn't get snapped off or broken. And instead it gets stretched basically and appears to us as then a thread that can easily distinguish a dogwood leaf, because all dogwood leaves have that. A couple more invasive shrubs. Here's common buckthorn at the top and Japanese barberry on the bottom. The sort of circular, somewhat orbiculate, opposite to somewhat sub-opposite leaves are a good characteristic for buckthorns. By sub-opposite, we mean they're almost opposite, but maybe not quite. These are opposite right here and here. It looks like there might be these two right here, just not quite exactly, so sub-opposite there. Buckthorns also have thorns, hence the name buckthorn, and the thorns are at the end of the twigs. And since the sharp pointy thing is formed from a branch, that's why it's called a thorn. The venation in buckthorns is somewhat similar to the venation in dogwoods. It has that somewhat arcuate venation pattern. So that would be, but one thing that helps separate buckthorns from dogwoods then is, especially this species of dogwood, the common buckthorn, is the sort of the glossy shininess of the leaves. Also, the fruits would be different on this species, as well as the flowers. In Japanese barberry, we can see it's a low woody shrub, and it has very distinctive sort of, you might call them teardrop shaped leaves, or obovate leaves would be another way of describing them. They're broader out towards the tip than they are towards the base, but they're all collected in little little spur shoots. So the leaves occur in clusters, and these clusters or these branches also have, I believe these are thorns that we see here. There's little short thorns that are present along these twigs. A couple more important shrubs are the autumn olive and Russian olive, shown here. So these have alternate simple leaves, and autumn olive by far is probably the most common of the two species. They're somewhat similar in having leaves that have sort of a silvery look to them because of what we see right here on the upper surface of these leaves right here. Some of those silvery scales is what those are. So there's a silvery look. It usually is a more dense on Russian olive than on autumn olive. But the way that these two are usually separated is on the basis of this characteristic right here. Autumn olive has brownish red scales mixed in with the silvery scales on the twigs and even some of the leaves. And that doesn't happen on Russian olive. Russian olive does not have any brownish reddish scales on the twigs here or on the leaves. They both have thorns, so that doesn't help separate them, but it might help separate them from something else. And their fruits are pretty similar. Some keys say that the fruits are somewhat different color, but that varies. And so you really can't use that. You're going to use the scales, the brown reddish scales here for the most part. Now there's a native species that looks a lot like Russian olive because it also is very silvery. That native species is silver buffalo berry, which is actually a threatened species in Iowa. So you surely would not want to be going out to Les Hills and cutting this down thinking you're cutting down Russian olive. It's pretty easy to tell silver buffalo berry apart from the olives, autumn olives, because in silver buffalo berry, the leaves are opposite. And these two species are alternate. And if you have the flowers, I don't have any flowers here to show you of silver buffalo berry. I have some flowers here of both of these two species. Here's the flowers of autumn olive, and here's some flowers down here. But in silver buffalo berry, when we compare it to especially Russian olive, the flowers in silver buffalo berry are smaller than they are over here. They are a more dull yellow color, not as bright yellow color, not as bright yellow as they often are in the autumn olives. And they're less tubular in their shape. But you really shouldn't have to even use those because you should be able to use the opposite leaves. Okay, we're getting close to the end. We've got, I think, just 3 more slides. We've got multiflora rose, which I don't think we'll just spend too much time on because it's pretty easy to distinguish from any of the other wild roses. That would be the important thing if you have the flowers, because multiflora rose is always going to have white flowers, whereas our native roses have some type of red or rosy red color. Of course, then the other important thing, in case you don't have the flowers, is you look at the stipules. Roses, wild roses, and the non-native roses have these. Here's a stipule right here. Again, it's kind of a different stipule though, because these stipules are fused to the lower part of the petiole. So they don't look like your typical stipules, but that's what they are. They're just kind of elongated and fused to the petiole. And that's what we're seeing here, that fused stipule, one right here and that one over here. But what's the important part here, of course, and we can even see it right here, is that the stipules have these little bristles along the margin, short little bristles. None of the native roses have that. They will have stipules that are fused to the petiole, but the margin of the stipules will have nothing on it. It'll just be smooth. This is an example of the prickles on multiflora rose, which are usually quite a bit more stout and definitely a lot more able to tear skin than the prickles on the native wild roses. And then down here we have another really bad one, Oriental Bittersweet. And of course, the problem here is trying to separate it from the native bittersweet. This is Celastris orbiculatus, and the native one is Celastris scandens. So I'm showing mostly, again, the Oriental one, and of course, one of the key things that's very helpful is the shape of the leaves. Usually much more orbicular, hence the name orbiculatus, or the scientific name here, versus the leaves on Celastris scandens, which are much more elliptical. There could be some variation though there, so it's sometimes it may not be completely clear, I suppose, just based on leaves. So there's a couple other characters I put in here that come from the Minnesota Wildflower website. One of them right here is showing that the flowers, if you have the flowers in the non-native orbiculatus, the flowers are arranged in clusters in the axils of the leaves. So you see a cluster of flowers here. There's actually one down here too, at a lower leaf, another one there, another one here, another one here. So all along the stem, the upper part of the stem, in the axils of leaves, there's clusters of flowers. On scandens, the native one, the flowers are all more compactly clustered together out at the end of the branch, at the end of the twig. Looks like there might be one cluster here in the axil of the leaf, but most of them are out here without any leaves. There aren't any leaves associated with these. The other one that's showing here is if you have the male flowers, and usually Celastris is dioecious, or have male and female plants. So the male flowers, of course, have the stamens. What this is saying is in Celastris scandens, the native one, again, the native ones have the pink borders, the anthers on the stamens are really bright yellow, whereas the anthers on the stamens of orbiculatus are more of a dull yellow or pale. Yellow. So you'd have to have the flowers, of course, for either of these. Some people say there's some differences in the fruits, but I don't see that as being very consistent. They can both have fruits that are reddish in color. Some people say that the fruits over here are more for scandens are more orange, and these are more red. But I've seen scandens have very red fruits too, so I don't think that works very good. All right, 4 grasses, and we're done. The first 2 are a couple of bromes. The first one I shouldn't have to talk too much about. If you know something about smooth brome, that's a real common, forage grass, roadside grass, a cool season non-native grass, perennial grass. The bromes are characterized by having closed sheaths. So this bottom part, this what we see right here is a sheath, which is the bottom part of the leaf. A leaf in a grass, it has 2 main parts. It has the sheath, which is attached to the node. All leaves have to be attached to nodes. So the leaf is attached to the node, and the first part of that leaf runs up the stem above the node as a sheath that surrounds the stem. And then when we get to the top of the sheath right here, then the blade becomes evident. The blade, which is the photosynthetic part, the flat part. Well, again, what we are saying here is in bromes, the sheath is closed, meaning that it's a solid piece of tissue that surrounds the stem that is that goes down through this here. The sheath surrounds the stem and it's a solid piece of tissue. In grasses that have open sheaths, the two edges of the sheath just overlap one another. They're not fused together to form a solid cylinder of tissue. Instead, the two margins of the sheath just simply overlap and can be easily pulled apart and separated. If you were to try to do that with a sheath that's closed, you'd have to tear the tissue here in order for that for you to expose that stem that's inside the sheath. So bromes all have that, and Japanese brome also has closed sheaths, just like all bromes. Japanese brome is an annual, and smooth brome is a perennial, of course. Japanese brome is a relative, a similar one called cheatgrass or downy brome, Bromus tectorum, are both very, very troublesome and somewhat invasive, especially in dry, sort of sterile soils. The way to tell Japanese brome, just vegetatively, I guess, is that from smooth brome here, for example, is the sheaths here are going to be a lot more hairy, just a lot of pubescence. And these hairs are usually pointed downward. I put this species over here. Canna brome is a native species of brome, a native brome that looks something like smooth brome, just a little bit like smooth brome. But you can see that the difference here, again, here's a closed sheath. The difference is that smooth brome has sheaths that are pretty much glabrous. There's no hairs. We don't see any hairs on this sheath right here. Here's another sheath here where I'm showing more of the sheath. I don't see any hairs really on it either. And the sheaths here have a very distinct amount of hairs here. Canna brome is a native species that's going to be found more in upland, fairly high quality forests, deciduous forests. So it's usually not going to be in a place where you'd expect smooth brome. However, if you have a forest that has some openings, you can certainly have a possibility that smooth brome could be in there, in those places that have a little more light. So it is helpful to be able to, of course, separate smooth brome then from canna brome, Bromus pubescens. And that pubescence is the best way to do that. The bromes are really separated into two groups on the basis of these things called the glooms. The glooms are the two basal bract-like structures in a spikelet, where a spikelet is the inflorescence structure of a grass where the flowers are. And again, this is a lot to try and explain in a short time, but these glooms, and here's some glooms here, and right here, there's always usually two glooms. One's a little bit smaller than the other. The point I'm making here is that the lowest gloom, which is called gloom 1, you look at it and you determine whether there's basically just one vein or if there are 3 to maybe even 5 veins. The number of veins on the glooms, the lowest gloom, separates the bromes into two main groups. Those that have just one vein, mostly, they might have two really short, faint veins towards the base, but they really may just have one complete vein that goes the entire length. Those are going to be smooth brome, canna brome, all the perennial bromes, plus Bromus tectorum. So Bromus tectorum, which is cheatgrass, that's how you would separate it from Japanese brome, because Japanese brome is pictured right here, and Japanese brome has 3 to 5 veins on that lowest gloom. So it goes into the other group. Also, very importantly, the smooth brome doesn't really have any awns on the lemmas. It can have a very, very short awn, maybe 3 millimeters, but basically no awns. Bromus pubescens has little awns that are 4 to 7 millimeters. This is Japanese brome. These are the awns right here, those slender threads. awns, and then again, they're coming off of the lemmas, which are part of the grass flower. The awns in Japanese brome are 8 to 13 millimeters long. And then the awns in cheatgrass, Bromus checkterum, are 10 to 18. So you can almost separate all 4 species just simply on the length of those awns. Okay, the last 2 species here are 2 perennial grasses. Reed canary grass on the top here. Again, a species you probably have dealt with quite a bit. And then tall fescue, another non-native cool season grass that is certainly a very important species in grasslands and a species you'd have to try to get rid of. Certainly if you've got it coming up in a planting, or it might even be present in a remnant pasture prairie. So first of all, reed canary grass, here's a picture of it, the large plants themselves. So both of these are going to have open sheaths, unlike the bromes. And the ligule is the other important structure then we're always interested in with grasses. The ligule on reed canary grass is a fairly tall membranous structure. We can see part of it right here. You can see part of it. I have to move this thing, I guess, so you can see it. Here's a really good look at it right here, this membranous structure right here. So a membranous structure that's fairly tall in reed canary grass. The ligule is anywhere from 3 to 7 millimeters or so tall, so it's pretty conspicuous. And I've noticed, I did a seed bank study where I had to check and identify reed canary grass at a seedling stage. And so I was trying to find a way to be sure that it was what I thought it was. I found that the lower part of the ligule on the outside, the part of the ligule that you can see, because it's this part here that faces away from the stem, that's the abaxial side. There's a fine pubescence right through here. You're not going to be able to see it very well here because it's really fine, but you can see it pretty well with a hand lens. And that's an important characteristic because that will help separate reed canary grass from another species that it could be confused with, a native species called Calamagrostis canadensis, a species that grows in sedge meadows. And it will have a flowering structure that looks a little bit like the flowering structure we see here on reed canary grass, but it has a membranous ligule too, and a pretty tall one and conspicuous one, but it doesn't have that pubescence. A couple other things here. Reed canary grass has leaves that are pretty broad. These leaves can be, well, let's say up to up to 20 millimeters wide, so 2 centimeters wide. And again, Calamagrostis canadensis, its leaves are going to be much more narrow than this. It only has, it's the length, the width of those leaves is usually no more than about 8 millimeters wide. So much more narrow leaves. Now the inflorescence on reed canary grass is much more open when it starts to bloom. It looks more like this. But when it gets done blooming, then it's more contracted and looks like this. And again, that's when it starts to resemble the flowering inflorescence of Calamagrostis. A couple other things. They both have small little spikelets like this, but in reed canary grass, what's happening here is that there's just one fertile flower right here. What we see here are some basically vestigial, sterile flowers. They look like basically like little hairs. That won't happen in Calamagrostis canadensis either. Calamagrostis just has one single flower. This one, again, essentially has 3 flowers, but 2 of them are degenerate, basically, and not fertile. Okay, last one is a pretty easy one once you see it enough and get used to it. Tall fescue, which is Festuca, was Festuca. It's now Pseudonoris is the genus name. Pseudonoris phoenix is the name now. Used to be Festuca arundinaceae. But anyway, here's how it looks growing out in the field. Very, very dense tufts of clumps, dense clumps of tillers. A couple of things with it are pretty characteristic. It has auricles, these little things that are kind of pointed right here, in association with at the top of the sheath. Here's the sheath again at the top of the sheath. Here's the blade. Auricles, they kind of come off the margin of the leaf here and wrap around the stem just a little bit. Also has a collar, often has a collar. That's this white area right here. That's much lighter in color, a light green coloration right here. And the leaf blade itself, dark green here, pretty dark green here, but notice how very conspicuous these veins are. These veins are really conspicuous, very kind of deep creased veins in the leaf blade. Another very good characteristic for it. Now, there's two Festucas, or Chinnonoris, I should say now, tall fescue and meadow fescue. And meadow fescue is going to look a lot like tall fescue. The way you separate them is you look at those auricles that again are wrapping around the stem at the base of the blade. In tall fescue, these auricles have little ciliate hairs on the margins. You can see them right there. In meadow fescue, which would be Chinnonoris pratensis, doesn't have those. The margin of the auricles is smooth. There aren't any little ciliate hairs present there. This just shows the entire spikelet of a Festuca, quite a few florets. Again, it can be more open like this when it's starting to flower, and then it can, the whole head kind of contracts a little bit more as the flower heads start to mature. So that is it. So again, thanks for joining all these sessions and hanging in here. We went over about 25 minutes here, I guess. It's fun to learn stuff as we can here in these workshop webinars, but believe me, it's always a lot better to learn stuff out in the field. And there's going to be quite a few opportunities for field trips. Just go to the Iowa Native Plant website. They've got a nice calendar there that lists, I think there's 3 or 4 Iowa Native Plant Society web field trips that are planned for summer. There might be some planned for Iowa Prairie Network too, but hook up with one of those field trip leaders, and that's a really good way, of course, to see plants in the real flesh. Any questions?
Yeah, we do have a couple here. One asks, are thorns on as many natives as invasives?
Well, yeah, there's thorns on quite a few native species. But as I'm thinking about it now, I'm just kind of running down a list of species in my head. I think there might be a few more, if you were trying to do some kind of percentage at least, possibly more thorns on a higher percentage of non-native species. Now I think a little bit more about it. Yeah, you mean to answer that, the best answer would probably take, I'd have to do some checking, run down a list of species, but I'm just thinking now, just off the top of my head, I can think of quite a few non-native species that do have, well, if you're thinking thorns or spines, that have thorns or spines, they're both, again, functionally they're the same thing. What they're doing for the plant is basically the same thing. It's just what they're formed from. And yeah, there might be some reason for that.
Okay.
Anything else?
Another one is, do invasives generally grow in the same area as natives? For example, sweet clover and purple prairie clover.
No, most of the time the invasive species are going to probably be coming into a community and getting established in some kind of disturbance. someplace where something has happened to kind of disturb the soil and maybe stress the native vegetation a little bit there, and just give them a better opportunity. So most of the time, again, you're probably going to see them more localized to places where, again, that opportunity for establishment has been better for some reason. Something's disturbed the soil.
Okay, and then let's see. Oh, do you have any suggestions for resources or places to go to learn about how to get rid of these species?
Yeah, we didn't cover that at all. And that is a huge important topic, and actually was the topic of one of the workshops at the Iowa Prairie Network meeting, a couple of weeks ago. Leigh O'Neill did a great job with Herbicides and Invasives 101 was the title of her presentation. That was not recorded, unfortunately, so there's no recording of it. The best way to start dealing with the strategies for dealing with these invasive species is probably to talk to someone who's got some knowledge, because it really is a lot of experience that is important. All land managers learn a lot about what works the best from experience, from trying things and seeing it doesn't work and then trying a different strategy. I mean, there's lots of different things to think about. There's lots of different questions to address, not just the herbicide. And of course, that's an important one if that's what you're going to use, but when do you use it, and maybe in what combinations, what timing are you going to use it? And herbicides aren't the only thing, of course. There's other strategies that can be used too. So herbicides are certainly a very important one, because most of these invasive species, the reason they're so successful is because they're very hard to control. And so, in most cases, it's going to probably take a combination of things. It might take herbicide even plus other things like mowing and fire, for example. Just a quick story there is that I'm doing research on a sedge meadow in Madison County, to the northeast of Winterset. I've been doing it for about 3 years now, and I'm looking at the best way to deal with weed canary grass there. And I'm trying combinations of herbicide and fire and mowing. And basically what I'm seeing is that it is the best thing that's working right now, it seems like, on our experimental plots. And again, looking at measuring the abundance of weed canary grass very quantitatively to get as objective data as I can. What it looks like the best thing there is a combination of a spring fire, and then following up with clethodim, an herbicide that is mainly kills grass. Doesn't kill sedges, and so that's why we're using it there, because there's lots of sedges on a sedge meadow. So yeah, that's just a quick example. There's lots of research out there, so if you really wanted to get into the scientific literature, you could answer some of those questions yourself by doing that. But again, another good way is just to talk to a land resource person who's been doing land resource management and using and fighting invasive species, for at least 10 years. Find someone that's been doing it for at least that long. And you've got a good person there that can help you a lot.
Sounds good. Well, I think that's all we got. Thanks everyone for joining, and thank you, Tom, for all of your work this winter.
Yeah, it's been a lot of work, but I hope it's useful for everybody, and hopefully, again, it's been worth your time. I guess that's the most important thing. If it's been worth your time to sit here and watch these things, then you've learned something. That's been the goal.
Yep.
Be out in the field.
Yep. See you later. Thanks.